Sorry, here. So we have done yesterday till this step. Like we had the raw FASTQ sequences. From their HQ FASTQ we received by using different tools. Then we assemble the genome into the draft genome yesterday. Right. From there we can do the genome annotation. So for the genome annotation, there are different tools available like PROCA, CRAST, CAS, PGAP. CARD is for the antibiotic resistance database based analysis for the search of antibiotic resistant genes in any genome or any particular sequence. Then we can also use the anti-smash database for the annotation of sequences like the secondary metabolites, like antimicrobial compounds which are produced there, or we can also use for the prediction of different secondary metabolites using the anti-smash database. All right, so we will do today annotation of the genome here. So what is particularly genome annotation is, when we have a sequence here, suppose this is a genomic fragment or genome sequence we have. So we can identify the genes and their functions. So related functions can be identified once we identify the genes, what genes are present on this genome there. So that the process of assigning the functional genes on any genome, that is called the annotation, along with the rRNA genes, and then the different types of the tRNA genes are also involved in the annotation process. Right. After that, what we can do, we have suppose this genome which was annotated, and then we can reconstruct the complete metabolic pathways which are present in any organism. We can also identify the different transporter proteins which are present there on the surface of the cells. So they all are encoded by the genome. So those sequences can help us to reconstruct the metabolic pathways there. Right. So different tools are available like the PROCA, RAST, CAS, PGAP, and I told you two more databases which are available like anti-smash and then the CAR database. There are other databases and the tools also available. So this is not only restricted to these ones. If you know some tools more than these ones, you can also use. Right. So in nutshell, what is the PROCA pipeline? Here in the PROCA pipeline, we give the input of FASTA contig sequences here. And then there are different tools which are implemented inside the PROCA pipeline. There, Aragon for the tRNA analysis, RNA MMER for the rRNA analysis, Infernal is for the non-coding RNA, Prodigal is used for the identification of the CDSs, and then signal peptides are predicted there using the signal P. From there, the CDSs which are predicted, these CDSs can be used for the blast search and then the HMER search, right? HMMER search. These databases are utilized, like the user can also use their own customized database or the database which are provided by the PROCA pipeline. So that will give us the protein annotation. Along with that, we can use the PFAM and then the TIGR database there for the protein domain identification and prediction. The output of the PROCA pipeline is the GFF, GBK, ASN format of the files, right? So we will also get the amino acid sequence files and the nucleotide sequences for those particular genes there. Right, so this is for the PROCA pipeline. There is another tool I told you. I will explain it to you how you can use it. So to install the PROCA, you can install easily it in the Anaconda distribution. So you can use the Conda install, and then these codes you can use there. I will copy this and send it to you. Right. There is a RAS server also available, so you don't have to do anything like standalone or you have to download. This server is available from the web page. So you can access that page from the rast.nmpdr.org website. From there, you can upload the sequence file like the complete genome or the draft genome, as we have the contig sequences with us, so we can upload that. But before to do that, we have to create an account on the RAS server. Once the account is approved, you will receive the email that your account is active now. Then you can log in it with your password, what password you use there to create the account. Right. So this is the pipeline where you submit the genome there, and then you get the annotation. So this all is the pipeline. They're like it finds the rRNA, tRNA, and then repeat regions are also identified. Then pyrolysyl and selenoproteins are also predicted there in this pipeline. Then it calls the CRISPR sequence or CRISPR elements, and then calls for the ORFs there. Annotate genes with k-mers, so fig fam and annotation or annotation base. So what is the annotation? Annotate hypothetical proteins using related genomes. So genomes which are available in the RAST database, seed database. They are used for the annotation process here. Right. Remove overlapping features and then give us the output. So the output can also be downloaded from the RAS server in the form of GPK file, GenBank file, and also in the amino acid sequences, nucleotide sequences, all we can download from the RAS server also. Right. The other one is the CAST server. So this is the KEGG automatic annotation server, as we don't have the access to the KEGG database freely. So we cannot access it without paying money. So, but the CAST server is still available free for everyone. So what it does when we give the input there, so that it could be the nucleotide sequences of the genes or the protein sequences we can use there. And it uses the different BLAST or the alignment tools like BLAST or the host search. From there, we can identify, it identifies based on the data, KEGG database, that what genes are matching with the database sequences. And then it gives us the KEGG orthologies. And if you're not aware about the KEGG orthologies, those KEGG orthologies are the particular set of the genes which are involved in the different steps of the pathways, metabolic pathways. So we can reconstruct the complete metabolic pathway from there. Right. So this is used for the reconstruction of the metabolic pathways there. Then the PGAP pipeline. So this is available from the NCBI, National Center for Biotechnology Institute. That is for a prokaryotic genome annotation pipeline. So a user can submit the genome to the database using the submit link here, right? So which is also available from the NCBI website. If you open the website, you will see the submit link there. Then you have to log in your account. So if it is not available, you can use your Google ID or Google. You can link your Google ID to the NCBI website and then log in there. Once you are logged in on the website there, you can start the submission process. So you can submit the quantic sequences, and then this PGAP pipeline will do all the tasks for you. So it will do all the annotations, and then if it finds any errors there, they will send you the email, and then finally they will approve the annotation there. Once the annotation is approved, you have to select a date to open it for the public domain. It will be available for everyone. So that is the one disadvantage of using the PGAP until you publish your data. But you can submit it and then use it. You have to submit it before the publication. So this is the best place to submit your genome. That is the PGAP pipeline, which is available from the NCBI. Right. So this is the PGAP workflow, like you submit the genome here, and then it does use the different types of the tools and softwares where it gives you the output there. So once the annotation is complete, you will see the link to download all the data from there. Right. So do you have any queries about this pipeline? One more tool I will show you here, the pathway tools today. I will not go much into the detail because you have to obtain the license from there, but that is one of the best tools which you can use for the annotation process. So where you can reconstruct the metabolic pathways, and also you can identify whether the pathway is complete based on the availability of the genes there, right? So you can do that in pathway tools. Do you have queries, questions? You can ask me.
Get a bank file. How can we convert that into a faster sequence?
So that alignment you want to convert to faster sequence.
Faster sequence, yes.
You can create the consensus sequence there, right?
Yes, sir. How, sir?
Using the SAM tools.
Okay, right? So SAM tools.
Yeah, you can use the SAM tools to do the alignment and then get some consensus sequences from there, right?
Can you say something, sir? I mean, SAM tools, which code we have to use?
here it will use
Okay, so samples first up.
yes.
Yes, okay, no, thank you.
I think this is to get the reads, not the sequences, right? So there must be in markup file. Okay. Anyway, I will tell you how you can
Yes,
extract the consensus sequences after the alignment.
okay, thank you.
I'm just missing it somewhere. Anyway, so once we have the context sequences, as we have analyzed yesterday, those two assemblies, defined and the default one. And I found that defined assembly was better than the default parameter camera-based assembly. So I selected this one here for the annotation using the Proca, and we will also upload this to the Rust server. To do the Proca annotation, we can use here, sorry. So I told you how you can install, like you can install the Proca, Conda activate first. Then you can create the env there for the Proca. So you can write here Conda create env, create -n Proca, right? Like this, you can create the env. Once the env is created, you activate the env, Conda activate Proca. It will be activated. Then the next step is installing Proca. Conda install -c. So different channels are involved there. So you can use like these channels here. Conda force, Bioconda, and default. Right. So Conda install -c Conda force, Bioconda, and default. And then finally you write Proca here at the end. It will install the Proca in your Anaconda environment, that particular environment which you have created. But remember to activate the environment before installing it. Right. Once it is installed, you can start using it. Other way to install Proca, that may be another process. Let me show you. You can run sudo apt install Proca. Right. You can install directly in your system file there in the user bin or user local bin there. It will install it there and then you can start using it. So once it is installed, we can run Proca -help. Right. So here, no options are required, but you have to give the input config faster file. After that, you have these different options available. So you can check all these options and depending on your requirement, you can use them. So first is the out directory. So output directory, it will store all the results in this directory here. Force, if the directory exists there, it will override the output on the existing ones. Prefix, you can write here to create the file names as the prefix. So you can write here like mycoplasma with prefix. So it will create all the files with the mycoplasma. Add genes in the GenBank file. So gene features for each CDS will be added in the GenBank GBK file there. Add mRNA also in the output. Locus tag, you can define here your own locus tag. So otherwise it will use that by default random locus tag, it will define, add there. Increment of the sequence number. So these counters will start at 1, or you can use like increase the number by 5 or 10. So it will use the first number as 1 and then next will be 10 like that, or 5 and then 10 and 15 like that. So you can change the increment. GFF version which is available here, default is 3. So that is good that you create the recent GFF file output from the program. Compliant here is for the add genes min config length 200 and center XX. So then if the names are not correct, you can use this. Config names are not accepted by the proca, then you can use the compliant mode here. Right? Center ID, you can use this here to add in the output file. Access and version to put in the GenBank file. Then we can use the genus, species, strain, and the plasmid sequences if the plasmid identifiers are there. So we can use here name of the genus, which genus it is, species, and strain names to include it in the output file, in the GenBank output. Annotation process will be based on these different lineages. So if you are, you know that this is the kingdom, or so it is archaea, bacteria, mitochondria, or the viruses. So by default, it uses for bacteria. But if you know that it is archaea or viruses, you can use the kingdom here and then define the viruses there. Genetic code we can use. So genetic code for different organisms, there are different genetic codes available, like 11 is by default. So genetic code here is the, for mycoplasma genome, what we are going to use here. So that will be 4. So we can use a translation table for the mycoplasma, that is 4. Gram positive, gram negative, these in this information we can use there. If you use genus specific blast, then you can use this. So you have to provide the genus here, right here. This will use this genus to do the genus specific blast. Then there are other options like protein, FASTA, GBK file to use the first priority here. HMMs can also be first annotate from the HMMs. So you can provide a trusted HMM and then the trusted FASTA file for the protein sequences to annotate from. E-value cutoff is 1E minus 0.9, right? So you can select here, like you can go much lower E-value for the 1E minus 12, 15, or 18 if you want more stringent annotation here, annotation output. Coverage is 80% here. So 80% will cover the protein sequence, otherwise it will not annotate it. So you can change this parameter also. And rest is the computation, so number of CPUs and then fast mode or the other modes you can use here. All right. So to run here, simply we can run this command. Proca output directory out dir proca annotation. Prefix is mycoplasma here. That is, the output file names will be created using the mycoplasma name. Add mRNA, add genes in the output files. Genus is mycoplasma, species pneumonia, and strain name UNK1. You can use here G code, genetic code, which we saw here. So that G code is 4. Here. Translation table 4 will be used here. Number of CPUs, and then minimum config length we want 200 base pairs. And then the input file name will be here. FASTA file, which we have for the annotation, right? It will do the annotation and then give us the output. I'm not going to run it because it will take like 7-8 minutes, so which will waste the time for us. I have already ran it. Proca annotation directory is there. So the output will be here inside this directory. Once the run is finished, you will see the log file for the errors, and then the if any problem has arisen there, you will see all those informations included here, written here. The output contains here the FNA file. So this FNA file is the sequence files for the 200 base pairs. So the smallest sequence will be the 200, or longer than 200. Not smaller than 200 because we use the filter there for 200. So this is just the config file, right? Then there is the FAA file here. This is the first amino acid file in the output. We see here, this is the locus tag, which was randomly assigned here, and then the sequence number. This is 0001, MGP protein, operon protein 3. So this was annotated as the MGP operon protein 3. Then the hypothetical protein. And if you open the next file, which is the FFN, so that is the corresponding sequence, nucleotide sequence file for this FASTA amino acid sequence. Here. So these are the nucleotide sequences for the same ID, 0001 and MGP operon protein, right? So these are the hypothetical proteins here. Same way, we have more output files, FSA, that is again here. That output, it includes the genetic code organisms, Mycoplasma pneumoniae and strain UNK1 for the contig sequences, right? So that is the same contig sequences, but with the information added here, like genetic code used, organism, Mycoplasma pneumoniae, and then strain name also included here. Next is in the output, we have the GFF file. So most of the times when we use the pathway tools or any other softwares to do the metabolic reconstruction of the organism using the genome, so annotation. So we can use, or sometimes we need to use the GFF file, or it is required to use the GBK file or GenBank file. So this is the GenBank file. So this GFF file is created also in the output, GFF version 3. So what it holds here, it holds the sequences. So this is the contig sequence name identifier, and then how it was annotated there, and then on from which range sequence number to which sequence number, then on which strand, plus strand or minus strand here. Then there are the ID. If you remember, that ID was written in the amino acid sequence file and also in the nucleotide sequence file there. Then the locus tag is also added here in the GFF file. So this information can also be used for different tools when you want to use it for the further analysis there. So this GFF output is also created. Text file is also created, so that everything is in the text format here. Like how many, what is the organisms, how many contigs were there, and how many CDSs, genes, mRNA, tRNA, tmRNA. All those informations are added here in the TXT. TSV is the same in the table separated, tab separated output file. So the same way, locus tag, gene, and length of the gene. Then what is the name of this CDS there? It is given. You might be seeing the GenBank output also. So you will be familiar, or if not, this is the GenBank file where it holds all the information about like the locus here, name of the sequence from where it was annotated, and then rest the sequence length here, how much is the length of this contig, and then the linear DNA. Right. After that, definition of the organism here, like Mycoplasma pneumoniae. So what we have added there, which is included in this output file. Then the source organism here, name, comment. Then you see the from which region, 1 to this is the total contig length, and then the where the gene 1 lies here. So this gene lies from 2 to 865, locus tag, and then the CDS here, locus tag of the CDS, then the next gene, and then their locus tag. What is the product name? So that is product is hypothetical protein for the one here. So MGP protein, operon protein 3. Likewise, you will see for the all of the other sequences also. So each contig is written at the bottom of the annotation there in the GenBank file. Here it starts for this contig annotation and ends up here at this position. So this is for one contig. It is for the all of the contigs if it is in the multi-FASTA format genome, right? If it is circular genome, we will see only single contig at the end of the annotation file in the GenBank file, right? So is it clear to you this proca run and then the proca output, how what we get here in the output? Is it clear? Yes, actually, I have included here for the prokaryotes, right? Okay, so once we have this output here, what we can do, we can go to other analysis. The other process which can be used here, you can use prodigal. Sorry. Prodigal is also installed when you install the proca. So if you install it using sudo or the conda environment, it will also install the prodigal tool. So prodigal is used to predict the open reading print CDS is there, right? It will identify the CDS in any sequence. It doesn't matter. We can also use the metagenomic assemblies there also for the annotation for the identification of the open reading prints. So if you're not interested in proca annotation, you can use the prodigal or you can install the prodigal separately, independently, other than the proca. So you can run a sudo apt install prodigal. Right. For eukaryotes, there are different tools available for the identification of the open reading prints. So once you have the prodigal installed here, you can run it and then you can create the protein translated sequences, protein translations, and you can also create the nucleotide sequences here. So, and GFF file can also be created here. So you don't need to run the proca there. You can run the prodigal and then create these files to use it for the RAST or the CAST server. So we can use the protein sequence files for the RAST and the CAST server to submit it, right? So we have the output here. We can go to the RAST server and I will submit this mycoplasma contig file, which is smaller than all the sequences which are longer than 200 base pair. So I will submit this one to the RAST server. Okay, so this is the rast.nmpdi.org, where you see here. Right. You can register for a new account, service or user group. So you can register here. You go to the register option, and then you write here all the information, login, email, organization, and then request. Once you request it, they will provide you the login username. It will be your name, what you want to use it. And then the password they will send you on your email, which email you will enter here. Once you are active on the RAP server, you can go to the here, right? And then login. So I'm using my user login and password. Right. So these are the genomes which I have submitted earlier. You will see all the list of the genomes here which were submitted earlier here, right? you can go to the top here and click on the your jobs and then upload new job. Okay. So you can select here the sequence file, the context sequence file you want to upload. We will use that particular sequence file, context define.pasta, but the sequences are smaller than 200 base pair in this one. So you can select the proca annotation. Inside the proca annotation, there is the FNA file, right? Here, this one, FNA. We will open this. Select this. So use this data and go to step 2. Click on here. Server is very busy because I saw that there was very heavy was written, that it means it's very crowded and lots of jobs running right now, right? So that's why it is behaving also slow. Maybe that could be the reason. So here you see the please enter or verify the following information about this organism. So if you know that this organism's name, you can identify the taxonomy ID. So where you can identify this taxonomy ID, you can go to the NCBI. And then you can look for the taxonomy here. Taxonomy database. Mycoplasma. And then the pneumonia. Search. I think the name is not correct here. Pneumonia is not right. Let's look for the mycoplasma. And then there will be mycoplasma pneumonia. I think it has been reclassified to some new organism name here. That's why I'm not able to see it here on the taxonomy. So that may be pneumophila. Something. Mycoplasma and then the pneumophila. Anyway, so if you have the organism, you can find it here in the list of the organisms on the taxonomy browser database. Once you know the taxonomy, so suppose this one is the organism. So I'm not using this taxonomy ID, but wants to show you. So this is the taxonomy ID you can use here, which is written for that particular organism. So this is the candidatus Macroplasma auti. So you can use this taxonomy ID to fill the RAST information here. Right. So for this one, this is 2104. It was 2104, but it is not showing there in the taxonomy. So I will find it out with where it has been reclassified, because lots of bacteria and the organism, this prokaryotes has been reclassified with the new names. So that may be the problem. So this is the old one. Bacteria, Terabacteria, and then the mycoplasmoids here. Yeah, so the name is now changed to Mycoplasmoids pneumoniae with this one, 2104, right? It is no more the mycoplasma. Mycoplasmoides pneumoniae, right? Then the genetic code for this taxonomy is, which is selected as here, 4 for the mycoplasma, spiroplasma, ureaplasma, and fungal. If you use the 11 for the archaea, mostly most of the bacteria, except these ones, viruses, and then the mitochondria, right? Use this information. If you want to use the strain name also, you can use it. UNK1 or whatever the strain name you have, you can use it. Then at this step, we can use the classic RAST or the RAST TK. So the updated pipeline is the RAST TK. We can select the RAST TK by default. And then you can use the customized RAST TK pipeline if you are interested in changing these parameters, like minimum identity, minimum length of the sequence, and all those other informations like minimum training length. So these you can select here to change the parameters. You can modify that and then use it. So I'm showing it, I'm using it at default. Then you can use the fixed frame shift, build a metabolic model if you are interested in, click on yes, and then turn on debug. If there are any errors, it will give you the error message there in the output. Set verbose level here. So set this to verbosity level of choice for error messages. So how the error messages will be shown there, you can set this like 1, 2, 1 or 0. Disable replication. So if this job is identical to a previous job, run from scratch. So you can click on it also here. Finish the upload. So the database which is used here in the RAST, that is the seed database, and then the genomes which are available publicly in the RAST database. They will be used for the annotation. So once it's submitted, we will see the how the process is running, like at which step it is, and it will show the green sign there that this step is completed, and then the next step is running, like that information will be given there. After that, we have to wait until it is finishing the annotation process. Till it finishes here, I will go to the KEG or the task annotation here. So this is very simple to upload the file here. You can use the BLAST option or the host X or the host Z option for the amino acid sequences. You can use any of these alignment methods here. Then the query sequence you have to provide here. In multi-FASTA, that could be protein sequence. So you just select here, choose file, and then the FASTA amino acid sequences we can upload here for the protein sequences. Open, right? You can write the query name here, plasmoides, right? Then you have to provide your email address here. Right. Then you can use here genes data set. You can use it for genes. So it will use all the representative organisms which are available in the list of this annotation database. If you want to use the for eukaryotes, you can use the eukaryotic organisms here. For prokaryotes, it will show the use the prokaryotic genomes which are available. Then organism list. You can select the organisms from the list also. So these are the organism lists, and then you can select the particular organisms to involve for the annotation. So you can select for prokaryotes, and then bidirectional best hit, right? So this is for the complete genomes. If you have the partial genomes, you have to use the SBH, single directional best hit. So the draft genomes and then the near complete genomes can be used with the bidirectional best hit method for the annotation there. So we can click on the compute by using the BBH method. Now this has been job has been submitted, but you need to confirm it from the email. So you can open your email. And accept the job. Right? Here you can submit the job or cancel the job. So you just click on here, submit the job. Then on this page, you will see the link to download the database, result here. So it will show you how the process is running, Mycoplasmoides computing, right? And once it is finished, you will receive the email that the job has been finished. And it will be kept for, I think, 7 days on the server, so you can download the result there. So let it finish, and I will show you how you can download the result from the CAST server. Once this is submitted on the RASP now, so that job was submitted. You can check the status of this submission. So you can view the status of your project on status page. Right. So here you can see this is the job, Mycoplasmoides pneumoniae, and then number of contexts, size, and then creation date, and then annotation progress. So you can see here that it was submitted but not started yet because job load is very heavy. So lots of people, they are using this server right now. They have submitted their jobs, and there are 406 jobs in the RAST queue. So it will take a while to start the job and then finish it. But I have earlier submitted some jobs here, so you can check one of them using the list here. So you can go to once the job is finished, we can click on the view details, and then it will show us the all information here, like how the analysis was performed. We can click on the browse annotated genome in seed viewer, and then the available download for this job. So that once the job is finished, you have the annotation. Now you can go to the GenBank. Sorry, this option here for the download, and then you can select the GenBank file if you want to download, or you can download the GFF or the amino acid or the nucleic acid FASTA file. So you can download any of these different formats from the download option here. Right. Share this genome with selected users. So you can share this also with some user. They can have the access on the RAS server. Close strains for this job. So what are those strains which were used for the annotation with this particular organism? You can see the list of those organisms here. Here you can see like E. coli, Bacillus, Escherichia, not coli, Bacillus, Synechococcus, Hermatoga, Mycoplasma, different genomes were used, Ureaplasma, right? So these many different genomes were used for the annotation of this particular Mycoplasmoides pneumoniae, right? So we can go to the browse annotated genome in the seed viewer. Right here, you will see the annotation output in the form of the subsystem information. So when we do the RAST annotation here, we receive the output in the subsystem features we see here. So inside the subsystem features there, in subsystem, these features are included here. So these are the subsystem features, right? And there are 24% of the fraction of the gene, this part of the genome of 24% are included in the subsystem feature counts. Right? And then the 76 are not in the subsystem, but they are also annotated. And 247 are hypothetical, non-hypothetical, and hypothetical are 363 genes. Here, 184 are non-hypothetical and 2 are hypothetical genes. Total 186. So less number of the genes which are classified as the subsystem features. So what are those subsystem features here? Like the cofactor vitamins, prosthetic groups, and cell wall capsule virulence disease causing genes, defense. Those genes are also available or not. Resistance to antibiotic and toxic compounds, right? Then DNA metabolism, secondary metabolism, regulation, and cell signaling. So those different cell subsystem feature counts are available here in this table. You can get more information about those genes here by clicking on them, right? So if you click on it, it will take you to the next page. Here, diagram is not available. But what are those genes? SSU ribosomal protein S12P. Right. Subsystem spreadsheet here. So this is the different genomes. Like these are my genomes which are submitted, and then the RAST database genome. Like active, yes. So what are those different organisms and how it is active there in those organisms? Mycoplasmoides pneumoniae UNK1. This one is that. And where it is ribosomal protein. If you click on it, it will open that particular fraction of the sequence. Here. Config number. So this is the sequence number, node 3 length, this one, and then run tool was used, this one. And you can see this particular diagram here for Mycoplasmoides pneumoniae UNK1, and this is that particular sequence. So you can get these figures also from there. Like SSU ribosomal protein S12P. Right. So this much of information you can get. This is a lot of information we can create here from the RAS server. So this is just in the subsystem features and then the annotations there. If we go back and again, we can go to the browse here. So browse through the features of Mycoplasmoides pneumoniae, both graphically and through a table. Compare the genomes you can select here based on the function, sequence based, and then the KEGG functions based also, KEGG orthology based comparisons you can also perform here. So you can go to the function based. Suppose we want to do the function based comparison. The list is empty. I don't know why, but it is not open now. right? So the complete genomes are available there also, and incomplete genomes are also available. Right. So these are the complete genomes which are submitted in or available from the RAST database. So you can select here any other mycoplasmoides. So we can select this mycoplasma pneumoniae, strain B, mycoplasma pneumoniae. This one. Select. So they are the same, I think. Select other. M129, right? So this is from the database, which is, you can select this one. Mycoplasma pneumoniae is strain 1M129. This one to compare with, and then you can get all the 296 which are in the subsystem features. So there are 296 in the subsystem features, and then you can see here the organism A and organism B. Which one is available? Yes, yes, and no. So this is not present in the organism A, and organism A for us is the our strain, and this is the database strain M129. Right. So this type of comparison you can also do, like which gene is missing. Like this one is not present. Find. So if you click on it, it will again try to find that gene. It is not certain that it will find it or not, but you can try to click on here and then take you to the try to find the gene there. Right. You can also do the same way for the KED comparison. So click on the KED here and then you can select the organisms to compare with. So this is done also. Annotation is also finished. It's taking a long time. So let me show you the result from the KED annotation. So from the CAST survey. So we see here the output of HTML and then the text output. Right. You can click on the HTML. It will take you to this particular page for the result page, where you will see like how many genes are annotated for each of the KED module. Glycolysis, gluconeogenesis 15 and 15. Then citrate cycle, right? Oxidative phosphorylation, tryptophan metabolism. So these are all the KED based annotation output here. If you click one of it, suppose you are interested in glycolysis. Here, so the green boxes which are shown here, they show that they are representing that this particular pathway gene or enzyme is present in this particular genome, right? and then the white ones are not present. They were not found. Right. So this is how you can try to reconstruct the metabolic pathways by finding all those different genes there. That is the one way to check. But this result is available for only 7 days. So what you can do the other way. Just click on text here. And this is the identifier locus tab. And then the KID orthologies for each of these identifiers. That they belong to this or they are this particular gene. What you can do, you can click on here, right click, and then save as this particular text output. Save it to the directory where you are working. Here, like give it a name and then save it. Now you can use this to reconstruct or visualize it anytime in future. So the next step you can use here, you go to the reconstruct KID pathway on the KID server, KID mapper. And that is the same output here, like the example files. If you click on this gene list.txt, this is the same output here. So we can use one of that, which we have downloaded. So choose file. You can choose that particular file here. This one, Genome JP Tools CAS. This file was downloaded from the CAS output as TXT. Select this open and then execute. Right. Now we can see that metabolic pathways 99, biosynthesis of secondary metabolites and other genes. You click on here. Suppose pyruvate metabolism you are interested in. So these are the genes and their KID orthology. Click on this pyruvate metabolism. You will see the reconstructed pathway. These green boxes are the genes which are present in this particular organism. Right. So this is the CAS annotation. The same way, this is not telling us that this particular pathway is complete or not. And if it is not clear to us that it is complete or not, so that is the problem for us. So to overcome that, you can also use the other tool here, that is the pathway tools, right, which is available from the bioinformatics.aisri.com, P-tools. You have to get the license here for the to use it. There are two different licenses available for the academic and then the commercial. So if you are from the academic institution, you can apply for the academic license. Click on the academic license. And then for the universities, colleges, non-profit research, government research institutions, and government agencies, they can obtain the license. So click on it, fill up the information, and then they will send you the link to download the pathway tools file. Once you have downloaded, then you can go through their video tutorial is also available. So you can go through how you can upload. So what data you can use here, data which we have generated from the PROCA, from the RAS server, and from the PGAP, from NCBI annotations. So NCBI genome database also contains the GenBank file. So those GenBank files, either they can come from the PROCA, from the RAST, or from the NCBI annotations. Those GenBank files we can submit to that particular this tool, and from there we can run it, execute, and then we can identify like this particular pathway is complete or this pathway is incomplete. What are those missing genes present there, right? So this is very useful tool also along with these what I have mentioned here, right? So with these few examples, I would like to wrap up today's session. If you have queries, questions, you can ask. I will try to answer. The link for this one I will share.